thank you for coming out on a really, really unpleasant afternoon. You've done us proud. This is actually the largest event that the Peabody has held since COVID, so by numbers. So give yourselves all a pat on the back. Really delighted to see you here. I'm Chris Norris. I'm the Director of Public Programs at the Yale Peabody Museum, and on behalf of Yale University and the museum, I'd like to welcome you to the inaugural John Ostrom Lecture. I'd also like to take this time to acknowledge, as we sit here on Yale's site, that Indigenous peoples and nations, including the Mohegan, the Mashantucket Pequot, the Eastern Peacock, the Scattercock, the Golden Hill Pagussit, the Niantic, and the Quinnipiac, and other Algonquian-speaking peoples have stewarded through generations the lands and waterways of what's now the state of Connecticut, and we honor and respect the enduring relationship that exists between these peoples and nations and this land. This is the first in a series of annual lectures that honor the memory of John Ostrom. John was a longtime curator of vertebrate paleontology at the Peabody Museum, an outstanding paleontologist, and a man who I think it's fair to say profoundly transformed our view of dinosaurs. And it gives me great pleasure to introduce one of John's successors, Professor Anjan Buller, faculty member in the Department of Earth and Planetary Sciences and a curator of vertebrate paleontology and vertebrate zoology at the Peabody Museum, to introduce this year's speaker. Thank you.
Well, welcome everyone again to the first John Ostrom Lecture, which I hope is a first taste of the new Peabody, which will be opening in early 2024 with galleries completely redone, but 50% larger than before, and it will be completely free. We've got new mounts of just about everything, a new K-12 education center. Can't wait to welcome you there. So, fossils are part of the stony fabric of our planet. They're a component of what we call the rock record. And in that way, they're geological, both in the compositional sense and the sense that they're studied by geologists. But they're also part of the book of life's history and are in that way biologic and can be viewed through a biological lens. To be a paleontologist is to try to straddle both worlds, often badly, always uncomfortably. But there's some who truly master this. O.C. Marsh and John Ostrom both were able to muster their knowledge of the living world to gain insight, to illuminate the lives of extinct animals. In fact, I think Ostrom's finest work was his work on the evolution of flight, which was so informed by the behavior of living birds, living bats. Ostrom had the great insight that birds are descended from dinosaurs, and his successor here at Yale, Jacques Gautier, who's in the audience today, showed rigorously that birds are dinosaurs, and that the characteristics of extinct creatures can be inferred from a particular kind of systematic examination of their living relatives. But no one in the modern age, in my opinion, has done more for the understanding of dinosaurs and other extinct vertebrates as living animals than Professor Larry Witmer of Ohio University. Larry is an anatomist par excellence, and that is perhaps, to me, the highest compliment one can pay a biologist, for anatomy is 3D, nay 4D structure, and is the basis of everything living things can do. Consider that even the twistings and foldings of DNA are anatomy writ small. The hardest problems in the life sciences and medicine have to do with biotic geometry, with space and time. Larry has his bachelor's from Cornell, master's from KU, PhD from Johns Hopkins. He holds the Cheng Ying Shin Professorship of Paleontology at Ohio, and he's published numerous groundbreaking, I would say discipline-creating papers in all the most exclusive scientific journals. He's appeared in National Geographic, CNN, NPR, media around the world. He is highly dedicated also to public education at all levels. His scholarly work has been a primary inspiration, maybe the primary inspiration, for my own lab's work here at Yale. And so it's with very great pleasure that we give him to you, for Professor Witmer is a living time machine. He will bring you as close as one can come to dinosaurs, not buried in the earth, but alive here before you once more. Larry, my friend.
Well, thanks, Anjan. I hope I can live up to that introduction. It is a tremendous honor for me to be invited to give the inaugural John H. Ostrom Lecture here in the glorious O.C. Marsh Lecture Hall, right next to the legendary Yale Peabody Museum. We all can't wait for that to open, and you should all be thrilled that it's actually going to be free admission. That's a wonderful thing. So the wonderful thing about John Ostrom is that I actually knew John Ostrom. I got to know him during my visits here to the Peabody Museum. I'd see him at conferences. I took this picture here back in 1985 when I was a master's student at the University of Kansas. John Ostrom has always been sort of a hero of mine when I was growing up, so it kind of blew my mind when I would go to a conference and he'd walk up to me and go, "Hi, Larry." I'd go like, "John Ostrom knows my name? And he wants me to call him John?" Just couldn't quite handle that. And it turns out that we're related, at least academically. John Ostrom here was the, did his PhD work under the mentorship of Ned Colbert at the American Museum of Natural History. Later, John was the PhD advisor for Peter Dodson, and later, who was in turn the PhD advisor for Dave Weishampel, who was my PhD advisor at Johns Hopkins Medical School. So that would make John my academic great-grandparent. And that's an amazing picture, except I really wish I were in it. There, I fixed it. You do what you can. So I'm not going to fully recount John's illustrious career, but I do want to point out why his work is so revered. Many achievements, certainly, many contributions. But he was a key player in two major debates, both of which have been so important and transformative for our discipline. One was the origin of birds. He did absolutely seminal work on Archaeopteryx, and he was a major player in the sort of the revolution in dinosaur physiology and behavior, drawing largely on his work on Deinonychus, a dinosaur he discovered and named, and which really is the model for the raptors of Jurassic Park. And so we're here to sort of, in a sense, celebrate John Ostrom's work. And so what I'd like to do today is show you how we get inside the heads of dinosaurs. If we're interested in dinosaurs as living, breathing animals, we do have to sort of look beyond the bones themselves to see what the bones can tell us about the soft tissues, the muscles, blood vessels, nerves, brains, skin that clothe and animate the skeleton, the features and the biology that time has stripped away. Really, ultimately, it comes down to how do we go from this to this, from a pile of bones to a fully functioning biological entity. And really, more to the point, what kind of science is involved in reconstructing the biological attributes of extinct animals? Well, where do we start? We start with bones in the ground, which we remove and carefully clean, and which we may mount in museums. We still kind of have a pile of bones, though maybe arranged in a more lifelike manner. In many cases, the bones themselves hide away their secrets within the rock in which they're entombed. And I call all of these dinosaurs here analog dinosaurs, because they live in real physical space, and you can touch them and smell them and taste them. And I love analog dinosaurs. But there's a lot that you can't do with them. And so we've made the jump into the digital realm, where you can take a skull like this T-Rex here and run it through a CT scanner at your local hospital, or in this case, one of NASA's rocket engine CAT scanners. And that step transfers that analog dinosaur into the digital realm. And once it's in a computer, you can spin it around and look at it from all angles, which is pretty handy because a lot of these guys get pretty heavy. But the real advantage in all of this is that you can look inside All of those secrets that have been kept away from our eyes for millions of years are now revealed. That dusty old pile of bones sort of starts to come alive, brimming, even dripping with biology. So, if soft tissues are so important, and dinosaur fossils are just a collection of bones, how do we get that soft tissue information that is so important? Well, obviously we need to look at animals that still have all of those soft tissues intact, and those are animals living today. So here are what we call extant animals. So here is dinosaurs, typified by T-Rex, of course, and where it fits into phylogeny, or the tree of life. Birds are most closely related. Birds are, in fact, a kind of dinosaur. And then things like crocodilians and turtles and lizards and mammals are more distantly related. But really, the present is the key to the past. It's kind of a slogan, but what it really means is that if you're interested in dinosaurs, you can't just look at dinosaurs. You gotta have one foot in the past, but you got to have the other foot firmly planted in the present. And so our methods run from the classical to the cutting edge. We use such time-honored, old-fashioned techniques like dissection and vascular injection and sectioning, trying to understand the structures of modern-day relatives of dinosaurs, really trying to give us ideas and information on the relationships of the soft tissues to the bones that we can then check for in the dinosaurs. And one thing I'll quickly point out is that all the animals, all the modern day animals that we get in are what are called salvage specimens, salvage carcasses that died somewhere else for some other reason or were euthanized and then were donated to our lab for research. But we also use the latest technology with CT scanning to get inside the heads of dinosaurs. CT scanning is an X-ray modality developed for use in hospitals, and we do a lot of scanning at hospitals. But many universities, like Ohio University and Yale, have high-resolution micro-CT scanners that are also very helpful for us. So CT is actually a wonderful thing if you want to visualize bones and teeth, the hard tissues. But one of the problems with CT is that it's not so great at visualizing soft tissues. So if we take this lovely little crocodile skull or head here and run a slice through it, you can see that all the soft tissues basically just wind up being a sort of indistinct gray. We need some way to enhance the contrast so we can actually see what's going on in 3D. So it turns out that administering iodine has been a game changer for the study of a lot of this morphology because iodine differentially binds to different soft tissues such that we can now see, it's not indistinct gray, we can see just about everything. It's really quite remarkable the detail that we can get. Well, we don't see exactly everything that we'd like to see because our lab is also interested in blood vessels, the blood vascular system, and how blood is shunted around the body. And we'll talk about this a fair amount as we go in today. So for example, what we do is we can take a carcass that was donated to us of a pelican in this case and inject its, we can use barium, which is a chemical that shows up on CT scans, and we can inject it into the arteries and veins and then subject it to CT scanning. So if we look at some slices here through this pelican, we can see that we've got nice bright white arteries and gray veins. So we can distinguish between arteries and veins. You can see that the muscle and the bone is much less dense and sort of a darker gray. If we look closer, we can actually see that we've got veins surrounded by arteries, as well as arteries within veins. That's really exciting for us because it opens up the possibility of having some physiological role here, particularly with regard to heat exchange and shuttling heat and cool around the body. Again, this is something we'll talk about today when we get to our dinosaurs. And so we've been using these techniques. For example, our vascular techniques, we're starting to explore the physiological significance of these using thermal imaging And so in this case, we were actually following up on the pelican example. We're able to actually show how blood that's cooled in the pouch of the pelican actually can be routed back to the brain to help moderate brain temperatures. And so these kinds of approaches wind up being important for us. And so this is just a summary slide showing the various kinds of techniques and visualizations that we can do. We still do lots of old school dissection. We do that all the time. It's absolutely essential. But these new techniques have been so important. We scientists often like to throw around the term transformative, particularly in our grant proposals. But CT scanning has really been transformative for the science of anatomy and morphology and how it functions. So, how can these techniques to help us understand soft tissues help us actually flesh out dinosaurs? Hollywood can just make stuff up. Looking at you, Jurassic World. But as scientists, we need testable hypotheses grounded in the structure of the fossils as well as their modern day relatives. Well, let's have a look and focus on this long-necked sauropod dinosaur, Diplodocus. So of course, we start with the fossil itself. But the fossils are often distorted, or they're missing parts, or they're broken, whether by nature or by human hands. What we really want is the actual pristine skull that was in the animal's head when it was alive, so that we can build the soft tissues on that. This skull looks pretty nice, and it is, but it's been living under a mountain for the past 150 million years and just kind of beat up. So what we can do then is, by CT scanning, we can pull that analog skull into a digital environment. And once it's a computer, hopefully we can start to fix it, to reverse the ravaging effects of time. That is, we can then use computer software like Maya or ZBrush to correct distortion, to replace missing bones, to fix broken elements, starting to bring that thing back into what we might call the skull that the animal had. And this winds up being a key thing. Now, you may quibble about any of the decisions that we made in making this skull, but given the vast variation within the natural population of Diplodocus 150 million years ago, I'm pretty sure there was a skull that looked pretty much just like this. In fact, this skull is really no longer CM11161. It's basically just Diplodocus at this point. And so now that we've got a complete skull, we can start to hang some soft tissues on it. So, so what we're going to do is sort of try to understand how we can flesh out this head. And now if you think about it, within our heads, there's sort of two classes of soft tissues that we have. We've got those that are sort of sitting within cavities, like our brain or the things within our nasal cavity. And we have things that span bones, like the jaw muscles. Well, let's start with the first one, things within bones, because those are a little easier. So we can take a skull like Diplodocus. It's actually a different skull than the one we just looked at. And we can run it through a CT scanner like we did here, and then digitally slice it, revealing a really nice cavity in the back of the skull that's filled with rock. We can then use software to digitally isolate or highlight, segment is the term we use in the business here, that structure that we now call a brain endocast. We then use the information we gained and experience gained from looking at the modern day relatives of dinosaurs to figure out what parts of that pertain to the brain versus nerves versus blood vessels. We can do the same thing with our modeled Diplodocus. And you can actually see this is, again, based on numerous specimens where we've looked at not just that one specimen, but every specimen we could find, constantly referring to things like the specimens, casts of them, CT scan data, photographs, the literature that allows us to restore these things, such that now that we can restore the brain endocast of Diplodocus completely, and we can actually extract it, view it from all angles, and even 3D print it, if we so choose. And so, if we think about some of these well-constrained structures, they include what we just talked about, which is this brain endocast in the back there. Also, some other pretty well-constrained structures, like a cartilaginous nasal septum, the same septum that we have, like somebody has a deviated septum, it's the same septum, as well as this air sinus right in there, and also portions of things like blood vessels and nerves that sort of lie within grooves on the bone, or actually tunnel through it in bony canals. But we also have less constrained structures. These are the harder ones to a certain extent. Like in some of the blood vessels and nerves actually pass through open space. Those parts are a little harder for us to reconstruct. Also things like the jaw muscles that run from the skull down to the lower jaw. Also things like the eyeball and the other contents within the eye socket, as well as the airway. And there's more things that we could add in here, and in fact are, but you can get a sense as to how all these soft tissues actually share space within the skull. Actually, in all of these ideas here, all of these soft tissues that you see here actually constitute hypotheses that we can test via a variety of criteria. For example, we use bony signatures, the osteological correlates. These are things like the scars that muscles leave, or the canals or grooves in the bones left by blood vessels and nerves. We can also use phylogenetics, and here we're referring to the genealogical relationships of organisms that tell us something about the expected anatomy. For example, we'd expect this air sinus in the front of the snout to be here because it's a shared attribute of dinosaurs and their relatives. So we would expect that to be there. And finally, packaging. Packaging constraints wind up being really important. The fact that it's all gotta fit means that all of the different soft tissue systems, in a sense, tell us about themselves. They tell us about each other. Sort of a process of reciprocal illumination. So using these kinds of things, we can start to flesh these animals out. A good example of packaging constraints are, in fact, the jaw muscles. And this work actually wound up being really important to us as we try to understand the function of the feeding apparatus in Diplodocus. Because as we were doing this work, one thing we discovered is that we actually couldn't get everything to fit. We had a problem, particularly with the jaw muscles. And so it turns out we made all these very careful and minute adjustments on positions, making sure they were right, but we wound up really having to adjust the thicknesses of the muscles, such that they wound up being very thin straps. And a key thing to remember about this kind of work is that in the real world, muscles actually bulge when they contract. And that actually winds up being a key factor when you're trying to do this kind of work, because in the real world, muscles can't be allowed to intersect or overlap. In the digital world, you can let things overlap. In the real world, that's not gonna happen. Also, what shouldn't happen in the real world, if a muscle bulges, it shouldn't cause the skull to explode. Important safety tip. So that caused us to adjust our thicknesses even further, which had some pretty significant implications for the biology of these animals. Just a quick aside here as a shout out to some of the team that was involved in helping us with the early phase of this visible interactive dinosaur work in our VID Diplodocus. This is about 10 years ago. And I'll point out that 4 of the grad students in this photo right here are now anatomy professors at medical school. And this woman right here in the middle, Kate Kirby O'Connell, is now an animation supervisor at Disney Studios. And so she's done amazing things. And so using these kinds of techniques, we've actually done more than just Diplodocus. And so we've actually restored more or less full head anatomy in a whole range of different dinosaurs. And we've got some others that aren't in this image. And I'll also point out and acknowledge the role of my longtime research associate and actually research collaborator on all this, Ryan Ridgely, and my good friend for over 20 years, who's helped me work through all this kind of work. And so all of these different, in a sense, to flesh out all of these things actually winds up being quite a bit of work. It's a very tedious process of iterative hypothesis testing and making all of these references to the structure of the bones and the relationships of the soft tissues, all those bony signatures. But the reality is we can do it. But what's really exciting is what we can do with it. That is, how we can use these restorations to start to explore things like the function, physiology, and behavior of these animals, what we call higher order inferences. And an obvious one is one that we just talked about, which is more realistic modeling of jaw and muscle movements, just like we saw here in Diplodocus. And I pointed out that our work on this sort of restoration of Diplodocus required us to have really, really thin muscles, which was very curious to us. At the time, Eric Snively was a postdoc in my lab, and Eric is really interested in engineering and engineering approaches. And so he wanted to have a look at this, and so what he did was he actually measured the lengths and volumes of all of these jaw muscles so he could calculate muscle forces. And what he found was that he was really interested in using an approach called finite element analysis, which is an engineering approach that tells us something about stresses and strains in skulls and actually tells us something about bite force. And bite force is a very important parameter for any animal, really. And so what Eric found was that the bite force of Diplodocus was actually just a quarter, 1/4 of what we thought it was before we did all this work. And so Eric's work has been very helpful, very important. Here is one of these finite element analysis illustrations showing an animation showing areas of high stress. And the hot colors show that where stresses are very high. And that's going to have a big impact on what it's going to mean for bite force. And so our studies showed that Diplodocus had a much weaker skull than what we had thought in the past. And in the past, we thought it had a pretty weak skull. And so these new findings impact our view on what the behavior and biology of Diplodocus must have been. Another project, again, this one was also led by Eric Snively, involved looking at restoring lots of the soft tissues in the head and the neck of a Jurassic predatory dinosaur called Allosaurus, based on a famous specimen known as Big Al. And what we used was borrowing another engineering approach, something from robotics called quantitative multi-body dynamics. And so we reconstructed, for example, things like the air sinuses in the snout and the attachments of the neck muscles, and basically ran all of these analyses. And cutting to the chase, one of the things we discovered is that Allosaurus's head and neck actually had something called a low rotational inertia. That's jargon, but what it means is that Allosaurus could move its head around with a lot of flexibility and control. We also found, it was an unexpected finding, that there was an unusual attachment of a neck muscle that indicates that Allosaurus may have had a sort of an unusual or different approach for removing flesh from a carcass, where it would actually use sort of a tugging action like we saw just there, something that we see that falcons do today, this tug or axial tearing, rather than something like a lateral shake of the head, which is what alligators do, and is in fact what our studies showed that T-Rex did as well. So we're able to use these kind of approaches to kind of drill down onto some fairly fine-grained aspects of feeding behavior in this case. All right, well, let's actually transition away from feeding biomechanics for a second and go back to physiology. One thing I mentioned before, that these vascular injections allow us to, in a sense, look at patterns of heat flow because the blood vessels actually shuttle blood around the body to warm and cool certain areas. This is something we know from our own experience. On a really hot day, we will become flushed. Our skin will be turned red as we shunt blood to the skin to radiate heat. Likewise, what we saw, just to remind you of our pelican here, where we discovered that there were venous blood that was cooled in the pouch was routed to the head to moderate brain temperatures and keep the animals from overheating. And so these vascular injection approaches really played into our dinosaur research in a big way with the work of my former doctoral student, Ruger Porter, and his studies of dinosaur vasculature and brain cooling. And here's the problem, and it's very well known. Dinosaurs were big. They were ridiculously big in many cases. What that means is that they had a low surface to volume ratio. And how you can envision that is like a big pot of stove or a big pot of chili on your stove. Large things heat up and cool down very slowly, as opposed to like these little dinosaurs you can see there on the right, which are sort of more like a cup of coffee that gets cold as soon as you turn your back on it, due to their high surface to volume ratios. So dinosaurs had these high heat loads. They would have been a big heat sink. And one consequence of that is that hot blood from the core would rush to the head and potentially damage the heat-sensitive brain and sensory tissues located there. And so, as a result, dinosaurs must have had some kind of physiological mechanisms to protect and cool those sensitive organs. We know that in modern-day animals, blood vessels form heat exchangers. And so Ruger's project was largely to try and identify sites of heat exchange in these dinosaurs. Just as the evaporation of our sweat on a hot day cools us down, animals use evaporative cooling just as we do. And so we targeted 3 main sites of evaporative cooling in the head that animals have, including the mouth or the oral region, the nasal region or nose, as well as the eyes and eyelids within the eye socket or the orbit. As always, what we do is we ground our inferences in the modern day relatives that we see here, and we did, I don't know, dozens and dozens of injections of modern day animals, in all cases trying to document not just the blood vessels themselves, but also their bony signatures, the osteological correlates, the grooves and canals that we can then check for in the dinosaurs so we can start to reconstruct dinosaur vascular anatomy as well. And so basically what we found is as in modern day species today, blood would have been cooled, venous blood would have been cooled in these same 3 sites of heat exchange, in the nose, the mouth, and in the eye socket. And sure enough, what we found were evidence of rich vascular plexuses in the nose and in the roof of the mouth, where venous blood would have been cooled by evaporative cooling and then routed back to the brain via a variety of different routes. And so this information showed us that Majungasaurus and Diplodocus, indeed probably more dinosaurs, maybe all dinosaurs, had the vascular machinery to help regulate their brain temperature, in a sense solving or addressing a problem associated with their large body sizes. And so although it looks like all of these dinosaurs probably had all these 3 sites of heat exchange, some dinosaurs or groups of dinosaurs actually emphasized different blood vessels. They had different thermal strategies. For example, something like Diplodocus and Majungasaurus actually seem to have a sort of a balanced strategy, meaning that all 3 sites of heat exchange actually seem to be important, whereas some actually emphasize one site or another. For example, Camarasaurus, the large-bodied sauropod dinosaur, really emphasized the mouth, the oral site of heat exchange, whereas the armored dinosaur Euoplocephalus here on the left actually did the reverse and was very much using its nose and nasal region as as a heat exchanger, an air conditioner. And so these kinds of approaches have been very, very helpful. As a brief aside, I'll point out that actually in 2020, National Geographic did a major roundup of dinosaur research, and they actually featured this project in a 2-page infographic right here. And here are our guys right here, is Euplocephalus and Camarasaurus and Majungasaurus. And so it was pretty cool that they did that. In fact, I really liked their title, and so I stole it for my slides. Don't tell them. It's cleverer than I am. Kind of a similar kind of a project was actually trying to understand patterns of airflow through the snouts of dinosaurs. Ryan Ridgely and I, a number of years ago, did a project where we pretty well nailed down where all the air spaces were within the snouts of a lot of dinosaurs. And in something like Euplocephalus, this ankylosaur, had a really unusual nasal cavity with all these twists and turns in it that ran right next to all those blood vessels that we just talked about, suggesting a potential role in heat exchange and potentially even metabolic physiology. We also modeled airflow with regard to the sense of smell or olfaction, as we did here in T-Rex. And we like to think this was an advance, but this was 15 years ago, and we were basically just drawing arrows. We're in a 3D virtual environment. We should be able to model it. And so actually Jason Burke, another former PhD student in my lab, wanted to tackle this project using another engineering approach called computational fluid dynamics. And I think our lab may have been among the first to actually start using CFD on animals. And Jason's been very busy over the years doing lots of modeling of airflow in lots and lots of different kinds of animals. And I'll point out that actually just about all of these are firsts. The first time that any bird has had its airflow modeled, or reptile or extinct animal of any kind. And if you look closely, you can actually see the air moving around in there. And it's actually telling us something about the speed. And one of the things that we can see here, that in all these cases, we get faster airflow in the main airway as it goes from the nostril down into the throat, and much slower airflow in the back part of the nasal cavity where smelling takes place. And it's been thought for some time that these slower air speeds in the olfactory chamber in a sense, allow the olfactorant molecules. These are little particles of smell. You might not think about smell as being particles, but they're little particles of smell that find their receptors. And the slower air speeds are thought to make that easier. And Jason's research, actually, his modeling shows that very, very nicely. So we wanted to apply this approach on an extinct animal, in this case. And again, Jason led this project. And this project focused on pachycephalosaurs, which are the bony-headed dinosaurs. In this case, Stegosaurus. Pachycephalosaurs are actually a great group to work on lots of things that involve soft tissues, because they ossified so many different kinds of soft tissues that other kinds of dinosaurs didn't. So if we take the skull and slice it in half, we can, of course, see this big, thick, bony dome on top of its head that gives these animals its name. But also what we can see is in the back part of the nasal cavity, right in here, are preserved unexpectedly a series of scrolled portions of mineralized cartilage. These are something you would actually call turbinates. Turbinates, because the scrolls sort of look like the veins on a turbine. And so they're in the region where smelling takes place, so we call these olfactory turbinates. One thing we also found was a ridge that runs right down the main part of the nose right there in the main airway that almost certainly had some kind of a turbinate attached to it. The key thing here is that ridge right there is in the main airway, and so any turbinate in there would be a respiratory turbinate. And the significance there is that respiratory turbinates have been sort of really thought to be important in metabolic physiology, because warm-blooded or endothermic mammals and birds have those kinds of structures, respiratory turbinates. So, turns out that we have all these structures too. And so we can sort of see the turbinates there in a skull. In life, those are actually covered with a mucous membrane, in which case we call those conchi. And these conchi and turbinates are actually involved in warming and humidifying and filtering the inspired air. But also, this is where a lot of the heat exchange is going too. Recall, this is one of those 3 sites of heat exchange, along with the oral cavity and the orbit. Now, the mucosa is, the mucous membranes are long gone in the fossils. That rots away quickly. But we might be able to get a handle on the turbinates. And so, that was what we set out to do with Stegosaurus. And we actually tested a variety of hypotheses versus a series of criteria. And I'll just focus on one criterion because it's sort of interesting, in that there should be some amount of olfactory airflow, some air going back into that olfactory chamber in Stegosaurus. And the reason for that is that we see that it's got really large olfactory bulbs in the brain. In other words, the neural part of the brain that's actually associated with the sense of smell. And again, there's also those well-developed olfactory turbinates. So there should be olfactory airflow. The kicker was, when we actually ran the analysis, in the absence of any kind of turbinate in that main airway space there, we actually had no olfactory airflow. In fact, the inspired air just completely bypassed the olfactory region, suggesting that there must be some kind of turbinate in there, if only to act as a baffle to direct air towards that olfactory region. So we can sort of look at the skull right here. Again, this is cut in half. And we do have evidence that we have that nice ridge right there that's very similar to the kind of turbinate ridges we see in mammals and in birds. And so that gives us the opportunity to, in a sense, build, well, actually you can see where that ridge right here is. We slice through the skull right here. You can see that ridge, that same ridge right in there in the fossil. And what we did then was to actually model in a turbinate attaching to the length of that ridge. We actually modeled multiple different sort of conformations or shapes of that turbinate, including being scrolled or being branched, which we borrowed actually literally from a turkey and an ostrich. It turns out that the branched conformation, the turkey turbinate, actually performed better. But the reality is that we don't really know what that turbinate looked like because we just have the ridge. But it turns out that with the analysis, it doesn't really matter because any of the conformations we used actually restored olfactory airflow. And so what we can see here is that the presence of the ridge gives us the general size and position of where that turbinate is, and the computational fluid dynamics analysis showed us that there must have been some kind of turbinate in that position regardless of what its shape is. And it's kind of interesting by modeling different shapes that the specific shape doesn't really matter that much. So there are things that we can't know, but what's useful for us to try to figure out is how much do we actually need to know. And so we know enough that there must be something else going on in there. And so in the publication, we talked about the physiological implications of this, what it might mean for warm-bloodedness and metabolic physiology. But we're also really interested in highlighting, at least to us, this notion that's kind of obvious in retrospect but overlooked, which is that turbinates also perform the function of acting as baffles to move airflow throughout the nasal region. So stepping back and just looking at this is actually an animal that lived during the Cretaceous, and we're doing some fairly refined knowledge or fairly refined studies trying to understand how these animals worked. And we can do that once we're in this digital environment. Well, let's go back to the armored dinosaurs, ankylosaurs, again. If you recall, I mentioned that Euoplocephalus had this long, really convoluted airway, sort of like the crazy straw that kids have, if you remember Crazy Straws. And so we were interested in actually what this might mean in terms of heat exchange, in terms of warming and humidifying inspired air, and at the same time recapturing moisture and heat during exhalation. And we had a great team to pull this together. Again, we had Jason Burke doing the computational fluid dynamics and Ruger Porter with his work on the vasculature. And this model right here shows one of the results of this work, which actually shows a remarkably efficient system for actually, in fact, warming the air and humidifying it on its way down to the lungs. So if we look at this slide here, which is a little complicated, this actually represents one of the ways that we actually assessed efficiency. In this case, we were looking at the efficiency of reclaiming heat during when the animal exhales. And actually, if we look at this image at the top left right there, that's basically what the fossil gave us. In a sense, that's the bony bounded thing. If we just fill it up and look at that, that's what the fossil looks like. And that's actually not bad. That's hovering around 70% efficiency. One cool thing about being in the digital environment is that we can actually generate anatomy that nature never did. And so this top middle image right there is one where Jason basically took out all the convolutions and made the nasal cavity much shorter, such that the airway went from the nostril down into the throat, just like we saw in Stegosaurus and just like in us. And you can see right there that the efficiency plummets to less than 30% if you do that. This one at the bottom is sort of interesting. This is one where we said, okay, well, let's keep all the length and we'll just get rid of all the convolutions, just make it a long, straight, narrow thing. And what we can see there is actually that's way better. It's at about 60%. It's way better than the short airway, but not quite what we see up with the fossil, the as preserved in the fossil airway, which that tells us that the convolutions actually make a difference. The convolutions probably relate to vortices or vortexes of air, and as they go around those curves, that actually apparently makes a difference. On the top right there, though, that's one where we actually went a step further and actually restored the soft tissues. We actually put the mucous membranes back in, and at that point, the efficiency soars up to around 80 plus percent right there. So these kinds of approaches allow us to, in a sense, do simulations and experiments within a computer environment that give us a glimmer into how these animals may have actually worked. And stepping back a little bit, one of the things that's sort of interesting is that ankylosaurs like Euoplocephalus don't actually have turbinates like we saw in Stegosaurus or people and a whole bunch of other animals. But what they may have actually by having this long convoluted airway is, in a sense, an alternate evolutionary solution to the same physiological problem. A lot of things ultimately just matter about surface area, an expanding surface area, no matter how you do it. Also, we can see how well Ruger Porter's work jived with Jason's work, in that we can see that these blood vessels, the veins would have actually been cooled by evaporative cooling as they're going past all of those, the mucosa within that convoluted airway, and then that cooled blood routes back towards the brain to help moderate brain temperatures. And I think John Ostrom would have loved all this. I think he would have, John was actually a geologist, but he was also a hardcore anatomist as well. And so going back to his doctoral dissertation in 1961, that was looking at these crazy crests on the heads of duck-billed dinosaurs like Corythosaurus, he actually did a lot of work with modern-day animals. So he was looking at things like jaw muscles and brains and airways in animals that are around today so that he could interpret his fossils. That's exactly what we do today. We've just got different tools. We've got more advanced tools to do that. And John's work has been absolutely foundational for work that we've done, sort of 21st century kind of stuff, looking at the structure and development of these airways and as well as the brain in Corythosaurus as well as some of its other animals. And this is a good sort of lead in to another benefit of this, these very visual 3D restorations, is that it's a really a good way to reach out to the public with our science because they're so visible and so easy to understand. Again, in the 2020 National Geographic, they did another infographic on our work, in this case of a cousin of Corythosaurus called Hypacrosaurus, looking at the airway, but also the brain and sensory systems. And also our work on Parasaurolophus, another cousin of Corythosaurus, will actually be appearing soon in a TV documentary devoted to some other things. And that picture at the lower right there shows me holding the 3D prints of the skull and airway of Parasaurolophus, which probably would have just been an unbelievable thrill for John Ostrom to have been able to do something like that. So in summary, the work of people like John Ostrom remains as vital today as it ever has. And we're just, in a sense, building on it. The modern technology has not replaced it or cast it aside. Rather, we're using it as a foundation for us to actually use these technologies to actually start to understand the biology of dinosaurs by basically putting them back together in a computer. And at the same time, that really gives us a new opportunity to reach out to the public and talk to them about science, just like we're doing right now. And thanks for your attention today. I'll just quickly point out, I took this photo this morning when I was working in the collections over in the Peabody Museum.
All right, thanks so much, Larry. So we will have a question and answer session for about maybe say 10 minutes now. And what's going to happen is I'll call on people, and if you wait until someone runs up to you with a microphone so everyone can hear the question, that would be great. Okay, let's go. Yes, over here, please. Coming. Yes.
Thank you very much. Just wondering, and it's not directly to do with what you're doing, but you must have thought about it. What about intelligence? Can you say with any definitiveness that certain species were more intelligent than others because of this and that?
Sure, yeah. The question was, what can we say about intelligence in these animals? And actually, I've been studying brains in dinosaurs for a long time. We've got a major grant that's looking at cognition in deep time. And intelligence is a sort of a difficult thing to do, and a lot of cognitive biologists prefer the term cognition as to how capable are animals of solving problems in learning and things like that. We colloquially would call that smart. And the reality is there are differences among dinosaurs in that capacity. We always tend to think that animals just need to be smart enough. And a key factor in that is that brains are very expensive organs. Brains actually cost a lot of energy that we take in. And so animals very often need to, in a sense, evolutionarily fine-tune their brain size so that they're not, in a sense, wasting energy on cognitive abilities or neural processing that they don't need. And so we see animals like T-Rex and other predatory dinosaurs tend to have relatively large brains because they're doing a lot of neural processing to be predators. Whereas something like Diplodocus, bless its heart, saw where I was going there, has really quite a small brain, but the dude was gigantic and it was just eating plants that aren't moving. And so its cognitive requirements were significantly less. And so the notion of dinosaur intelligence is something that our broader team, it's actually a multinational team, is actually working on. So thank you for that question.
Up here, top back. No, way back. Saw your hand up this whole time.
Are you like holding a Deinonychus over there?
Yeah, that's a Deinonychus. Same Deinonychus as John Ostrom right there.
Whoa.
Yep, and that's a Deinonychus foot right there with the killing claw.
For a second, I thought that was like a velociraptor.
Well, it's very similar to Velociraptor. Velociraptor in the movies is like this big. In real life, it was about this big.
Yeah.
Before about Hollywood.
I watched the franchise a lot.
You and me both, dude.
You can come see that Deinonychus when the museum reopens. It's here. Okay, let's see. How about the back of the front row here?
Do you use to determine how strong a jaw muscle to like help determine how strong a jaw muscle would be? Do you use whether they would be a carnivore or a herbivore? That word that I can't pronounce yet. Okay. Well, I normally can, but okay. Because the carnivores might need more jaw muscle to actually tear the flesh off, whereas the herbivores wouldn't need to do that.
Yeah, that's a great question. So the question was, well, what kind of muscle force does it take to be a predator or an herbivore? And the answer, as is so true in much of life, is it depends. And it depends on what kind of a carnivore you are and what kind of an herbivore you are. So for example, if you're T-Rex, an adult T-Rex, and you are biting bones, you need big, powerful muscles to actually crack through those bones. If you are Deinonychus, your skull is probably adapted more for removing flesh using other ways to kill the animal. And Deinonychus is actually a fairly delicate animal in many ways. And so it was probably not cracking bones. Something like Allosaurus. We talked about Allosaurus and how it was actually pulling back. And in that case, when it was removing flesh from the bone, it was actually just holding on with its teeth, and the muscles that were actually working were the neck muscles to pull that back. Same thing with some of the plant eaters. Something like Diplodocus right here, basically its teeth are kind of a joke. They're just these little, they look like little crayons that are jammed in the front of its face that were probably used sort of as rakes or something to strip branches off. They were not chewing. And so they had relatively like super weak jaws and bite force. Whereas there's other dinosaurs, other plant eaters that have really powerful bites. And particularly if there's animals that are doing things like biting repeatedly, like chewing, or cracking seeds, that requires big jaw muscles. A cardinal at your feeder has really big jaw muscles because it's cracking seeds, whereas a sparrow at your feeder doesn't, because it's just picking up little seeds and chucking them down. So it depends, but it's a great question. Thank you.
Oh, bottom here in the blue.
How do you figure out how to do all of that?
Yeah. That may be the best question we got. How do we figure out how to do all that? I spend a lot of time explaining that to that thing that says NSF up there. That's the funding agency, but that's a great question. I don't mean to make light of it, because actually it's kind of hard to do all these things, and there's lots and lots of steps. And you saw that I had a lot of people that I work with. I work a lot with graduate students and undergraduate students. I've even had high school students working in my lab and postdocs. And we work as a team, and we often do different parts of it. Like you saw, I had one person that was working really hard on blood vessels and trying to figure out where they went. Another person that was trying to figure out how airflow works. And so all of these team members get together and they work as a team. But it is hard, and it takes us a while sometimes to actually come up with these things. But thank you, that's a great question.
Okay. Lisa, how about right next to you?
You talked about Euplocephalus and its very complicated nasal passages. Now, in more recent days, I've seen sort of interesting speculative reconstructions of Euplocephalus and similar ankylosaurs with like nasal air sacs attached to the nostrils that will inflate and possibly like make a sound to communicate. Your thoughts on that? Is that plausible? Or?
Sure, yeah. So the question was, some people have actually suggested that some dinosaurs could have had inflatable sacs that are coming out of their nose, which sounds pretty crazy, but animals do crazy things. And so, I mean, what Euoplocephalus does without that is pretty crazy. So if Euoplocephalus had inflatable nose sacs, that would be maybe right in line for that animal. The reality is some of these things are pretty hard for us to get at. The person that actually presented the notion of that for, I think like Triceratops and its relatives, a ceratopsian, I'm not sure I think he was being a little whimsical when he did that. But the fact of the matter is, inflatable display organs are used all the time in the animal kingdom. I don't know if you've heard the frogs out yet, but you'll get the peepers, and they'll be actually doing it in many species. It's actually brightly colored. There are things like elephant seals and hooded seals that actually have inflatable nose bags that come off of their apparently the females just love it. No accounting for taste. But, and so could some of these dinosaurs have it? The answer is maybe. And those are the kind of questions that actually we are trying to get at. One of the things we've discovered over the course of time is that a lot of, and sort of in some respects, my basic job has become is where is the limit of what we can know? It turns out a lot of the really interesting things are like right out there. They're just beyond the limits of what we can know. What's interesting is that technology like CT scanning and all these other techniques are boom. All of a sudden we've got some new techniques that we can start to know more than what we thought. And also just making the right comparisons. Actually starting to look at, we've actually dissected hooded seals and elephant seals because we have actually are interested in that question. I have come to learn to never say never about life because life just evolves all of these different kinds of organisms. Thank you, that's a good one.
Okay, let's go, how about front and center here? Or at least I haven't run all around.
Thanks. So I guess something I'm wondering about, we know that dinosaurs, I think at least, became warm-blooded over time. I'd imagine from a heat exchange standpoint, those would be, kind of different heat exchange problems when you're dealing with a cold-blooded creature versus a warm-blooded one. Do you see any evidence of changes in sort of the heat exchange efficiency over time?
Well, it's so the question was, with the evolution of warm-bloodedness or endothermy, would that occur over time, and would we see changes in that? It's a great question. It's a difficult one, but one that we've thought a lot about. And often it's not so much over time, often it's within different groups. Different groups will often have different strategies. And so the reality is it's really hard to be warm-blooded like we are. I mean, it's actually hard to maintain body temperature at a peak rate, and there's lots of costs that go around with that. Like breathing fast. We breathe faster than cold-blooded animals. And that's actually a lot of these things that we're looking at in terms of like reclaiming heat and water. It's because we breathe really fast, and you can actually dehydrate yourself when you breathe too fast. And so we are actually indeed trying to look at those structures. What's interesting is that a lot of these, we think about hot-blooded dinosaurs, a lot of the things we're looking at are things that are because dinosaurs are too hot-blooded. In other words, endothermy makes some of these things worse. If you're a big dinosaur, like Diplodocus or any of these guys, where you're just a giant heat sink with the sun coming down on you, heating you up, you're going to be hot, whether you're generating heat internally, like a warm-blooded, an endotherm does. You're going to be hot anyways. And so these kinds of we call them physiological devices, things that you can use to, in a sense, deal with these heat problems, are going to be true for so many of these dinosaurs. What's interesting is we find evidence of a pretty legit nasal respiratory turbinate in this little Stegosaurus, which is right down here, which doesn't have the big overheating problem. If anything, that guy would get chilly from time to time when it was cold. And so there's a decent chance that something like that would have had, if it were endothermic, it would have had the machinery to actually help deal with that.
Okay, all right, we're getting close to past 10 after now, and with great apologies, I think we're gonna have to cut it off. Thank you so much.